Hi, I'm Tom Bumol. I apologize for not being with you in person today, but I want to thank the organizers for the opportunity to discuss the biotechnology strategy at Lilly as a key innovation path for our company. Next slide, please. In the late 1990s, we decided on a strategy that would have us look at reengineering biotechnology as an important source of innovation for Lilly. Today, we are the 5th largest bioproduct company by total biotherapeutic sales, and we've had many first-in-class molecules, including those shown in this slide, such as Riapro, Humalog, the first engineered insulin, Zygris for sepsis, Forteo, the first anabolic agent for osteoporosis, and with our partner Amylin Byetta. But there's been a discontinuous stream of innovation coming from Lilly Research Laboratories and our collaborators, and we made the decision in the late 1990s to invest in building a new integrated infrastructure that allow us to look at line of strategic sight from discovery through development, devices, manufacturing, and of course through to the patient. Next slide. What fueled our interest in biotechnology is the incredible amount of investment over the last 10 years by governments around the world, about private research institutions, and of course the pharmaceutical and biotechnology research and development engines that have really unraveled incredible opportunities, understanding new omics, proteomics, and the Human Genome Project, but more importantly, the new targets that this provided, new physiology, and we do believe that despite the hype of the late 1990s, that this incredible intensity of information, although complex at times, is translating to new pathways and new opportunities for intervention, and the pipelines are clearly emerging across the world, and for biotechnology product candidates. Next slide. We believe that a key innovation space for protein-based therapeutics lies in the fact that in our human physiology, cell receptors, hormones, growth factors, and antibodies play key roles endogenously in us. We have protein ligands talking to receptors for proteins. We have ligand antagonists and receptor antagonists as antibodies. And we believe that these fundamental designs of nature, of signaling and antagonism, can be mimicked by design for tailored therapeutics and biotechnology. We believe this will be possible through engineering proteins, peptides, and antibodies, and we think this is a strategy that we hope will provide a key opportunity for a number of new therapeutics, and we'll illustrate some examples in this brief overview. Next slide. Protein engineering, we believe, is key. We want to keep the patients centered in our thinking. Our design principles for this tailored engineered therapeutic are really looking at patient outcomes. And how can we design agents to be maximally efficacious by altering their pharmacology? We want to minimize the safety risks and maximize the safety and therefore improve the therapeutic index. And importantly, from the perspective of making proteins drugs, because nature didn't design them to be, to make them have drug-like profiles that allow us to safely manufacture them, purify them, characterize them, and deliver them to patients. And importantly, on the delivery piece, our commitment to really making biologic therapies very, very easy to formulate and have a very important and easy interface for the patient. We believe these features are key to giving designed therapeutics an advantage for the Lilly pipeline. We've made commitments in our alliances and our mergers to really make this type of possible approach available to our scientists and our teams across all of our therapeutic areas. We've also decided over the last 12 years, in the next slide, to really transition from a diabetes-centric organization to a therapeutic area integration across all of our therapeutic areas of interest. And this has included combining the extensive therapeutic area expertise that we would have in our therapeutic areas, such as neurosciences and autoimmunity, cancer, and endocrinology, with extensive and deep biotechnology expertise in discovery and development. If we keep the patients, as our final target, we think these diseases then become possible from a strategic line of sight, really taking exciting new concepts forward way beyond our historical presence in diabetes to now include these diseases, including Alzheimer's disease, multiple sclerosis, Parkinson's disease, in areas such as cancer, where targeted therapies have become extremely important in terms of combating the unmet needs there. And obviously in the areas continued innovation in diabetes, diabetic nephropathy, obesity, osteoporosis, and many others. Next slide. This approach over the last 13 years, plus the merger with Applied Molecular Evolution, where I'm currently based in San Diego, California, as well as the merger and acquisition of ImClone last November, has really established Lilly as a biologics company with significant pipeline. Shown here are some of the key assets in our early phases of clinical development, phase 1AB and phase 2 and phase 3, but more importantly, a very robust pipeline at the left-hand side of the slide, which discusses our pre-FHD, our first human dose preparation, which we have essentially 10 or 11 molecules poised to enter clinical development, including some this year and next year, to really keep building this in a sustainable way. We're excited about this because it takes advantage of these signaling molecules and the antibodies and provides real breakthrough opportunities for therapies across many areas, as some are listed here. Time does not allow me to go into all the areas, but I want to highlight some specific areas that are of keen interest to us in translating research from basically the research labs around the world to the bedside and clinical validation. Inflammation is an important area for us to consider. The science of the last couple years have really illustrated that what we knew about the different subsets of lymphocytes had to be rethought. Textbooks are being rewritten. We understand new pathways on how T cells, and particularly pathogenic T cells, are developed, what keeps them going, and this has led to subsets such as the Th17 cell, which we now believe are key to autoimmunity and led to our discoveries of several agents, which we'll describe in a second. Similarly, we see this on the B cell, which has led to interesting discoveries as well. And this has opened up several important possibilities, not only for autoimmunity and inflammation, but for related diseases, including things such as diabetes, as well as oncology applications of some of these approaches. Next slide. We've disclosed recently 4 important assets related to these pathways, and they're listed here on this slide. And I'm only going to describe one of them for you today, and that is the IL-17 antibody. Although we are extremely interested in a variety of other approaches, including partnerships with our partner Macrogenics on teplizumab for type 1 diabetes, as well as other collaborations ongoing within the organization. But IL-17 antibody in particular is interesting because this is one of our engineered human antibodies to a key T cell cytokine. We have a positive proof of concept clinically, which I'll describe, and currently advancing this in phase 2 studies in various autoimmune diseases. So how did we do this? We were excited scientifically, and just to give you a view of the process of these new discoveries, and some of our key scientists, immunologists by training, were aware of these breakthroughs. And we saw IL-17 as an incredible opportunity to block its function specifically. And the beauty of antibodies is that we can make selective, potent, high-affinity antagonists to essentially knock out that function in preclinical models. We were successful in doing that, and that gave us great encouragement to take this agent forward into safety testing and then ultimately human testing as we went from bench to bedside with a targeted therapy hypothesis. What also stimulated our urgency here is that the research continues to expand on IL-17. This interleukin drives the pathologies seen on this slide, which include effects on synovicytes, monocytes and macrophages in various diseases, endothelial cells, chondrocytes, the osteoclasts, the neutrophils, and essentially a pluripotential effect driven by pathogenic T cells in joint inflammation and in other autoimmune disease. That was incredibly intriguing. When you see a specific mediator that drives that much pathology, can you specifically block it? And can you see a result that really gives you the outcome you're really looking at? And that was what drove our work over the last couple of years. Our scientists, both in Indianapolis and in San Diego, began to make these primary antibodies. We engineered and optimized these antibodies, and then we went to clinical testing with collaborations across the globe, including Stanford and in Belgium, along with our Chorus group at Eli Lilly and Company. And we disclosed this particular abstract for LY243921 just a few months ago at the EULAR meeting. But it turned out in this safety and tolerability study in early phase testing, we actually saw a demonstration of positive proof of concept, which I'd like to share with you right now. What we saw after administering this antibody over the doses illustrated here at 2 milligram per kilogram, 0.6 and 0.2 milligram per kilogram is that in patients that were naive to previous biological therapy, that we demonstrated a very robust ACR50, which is a scoring system that looks at joints and tenderness and pain and other functions used by the American College of Rheumatology to generate a robust objective effect in patients. What was very exciting to us in this slide was that not only did we see a robust effect in these patients, but it was a very robust effect at all doses that was very similar or akin to studies that have been published related to the anti-TNFs. The anti-TNFs, of course, are very successful interventions, but this is a very different approach looking at a specific blockade of the Th17 pathway, and we think dissecting the immune response in a very specific fashion, which we hope will have advantages in numerous situations. This is just an example of how we can quickly go from concept, which can come from the literature, which can come from a variety of approaches and our own scientific research, to building an antagonist, making sure it's safe, making sure it's engineered to be very convenient for patients. And in this case, we just gave several injections and saw a durable effect over 16 weeks. So a very exciting new paradigm for thinking about rheumatoid arthritis therapies, and get to this positive proof of concept, which allows further investment. Don't have time to elaborate on this, and unfortunately I won't be there to answer questions, but it's just one example of many that we have of the 20 plus agents we now have in clinical development. Biotechnology is allowing us to go very rapidly from concept to proof of concept. And in business of innovation, that's absolutely critical because we can do all the preclinical research that we want, but it's the human studies, the human research that really give us confidence in improving probability of technical success, and importantly, the opportunity to really advance new medicines, which we think will be important for patients and for our customers. And the final slide, this has been an important journey for us. We believe that innovation is key to the growth and future of the biotechnology and pharmaceutical businesses. And we do believe that the biotechnology approaches, because of their specificity and their tailoring and the engineering capabilities we have, really allow us a unique opportunity to take new concepts into humans and make this vision a reality. Our journey has been particularly exciting for us for the last 5 or 6 years. We've integrated our biotechnology research organization with our therapeutic area organization. We've had the merger and acquisition of AME in approximately 5 years ago. We've committed to a development organization in Indianapolis that allows us to scale up these entities. And more recently, we've committed to multi-use manufacturing facilities in Ireland, and with the merger and acquisition of mClone, have a robust cancer portfolio to complement our internal cancer portfolio to really make this an exciting opportunity. Our strategy is on track. We think it's on track to meet annual launch goals for 2011 plus from this overall pipeline. And we now currently have over 21 biotechnology molecules in clinical trials, and really an exciting opportunity to extend, we hope, a new innovation approach to patients. Thanks for the time, and again, apologies for not being there in person, but hopefully we'll have an opportunity to discuss this with my colleagues.